Good morning, everybody. Welcome back to BIOL 1313 with Sol Ross, Zoology. Today we're talking about the cell cycle in meiosis and mitosis. So getting into that, one addendum I need to make to our previous lecture is I told y'all that humans have 23 chromosomes. That is not technically wrong, but it's certainly not the best way to express that. Humans are better described as having 23 pairs of chromosomes. These pairs, the two chromosomes in each pair, are known as homologous chromosomes, and in these they basically have the same genes, they do the same function, but one of them is from the mother and one of them is from the father. So there are two copies of each chromosome, one from the mother, one from the father. We have 23 pairs of homologous chromosomes, or strictly speaking, 46 chromosomes all told. So the cell cycle. So before getting into specifics, I want to just basically describe to you what I'm talking about when I talk about the cell cycle. So this here is a, there we go. So this here is a very kind of simplistic description or visual description of the cell cycle. You can read this as proceeding from the top left to the bottom right. So this is step 1, step 2, step 3, step 4, step 5. And yeah, so basically what happens in the cell cycle is that is the process of one cell becoming two cells. So what we see here at the top left is this is, just a cell. This could be a skin cell, stomach cell, who knows, just any cell. And then it goes through a series of steps. This step right here describes the basically the division of the cell interior into two portions, each one basically destined to be an offspring cell. And then you can see over here and over here, that's basically the process. You can very simply think of it as, splitting a bubble in half or something like that, just like a soap bubble. Yeah, so you start with one cell, end up with two cells. And that here on the bottom right is, oh yeah, that's a little bit behind my face, I think. At any rate, you can see this is two genetically identical daughter cells. And so this is the kind of cell division that allow us for growth and maintenance of an animal. So say, all of us, when we were born, we're a lot smaller than we are now. All those extra cells came about in this manner, basically with the production of genetically identical daughter cells. When you get a cut or something like that, the tissue or the skin that eventually is that heals that cut, that is also produced in this way. And yeah, so one important distinction here. So I say that these two daughter cells are genetically identical to the parent cell. So that's generally true. The DNA is ideally replicated identically, and so ideally the two daughter cells are identical in every way to the parent cell. That's of course barring the possibility of mutations, which do occur, but in theory, at least ideally, they would be genetically identical and typically are. So this is a graphic showing the cell cycle taken specifically from y'all's required reading. And so the cell cycle is typically taken to begin at the end of a process of division. So actually, let me step back here. Thanks. Where are you going? Okay. Yeah, so as soon as you get that bubble split, at the moment that one cell becomes two distinct cells, that's the beginning of the cell cycle, and then it goes all the way back around to well, basically you have one cell, you have two cells, then you take one of those two cells, this guy right here, and then the end of that next cell cycle is when that one breaks off into two cells itself. So there are two major portions to the cell cycle, each with many subportions. The two major portions are what are known as interphase and the mitotic phase. And interphase is basically the bulk of the cell's life cycle. Oh, yeah, basically the bulk of the cell's life cycle. That's when it's in the body doing its function. So say if it's a stomach cell, it's in there doing stomach things. If it's a neuron, it's in your brain, doing brain things. If it's in your skin, it's, in your skin. Um, so that's really so the main activities carried out during interphase are not really relevant to cell division, and so I'm ignoring all of the the metabolism and functions of these cells during most of their life. But we have to, of course, include this as part of the cell cycle, and specifically this portion right here, the S phase, is an integral part of cell division, but is considered to be interphase because it's basically, yeah, it's distinct from the mitotic phase, which is over here, but there is a major period of DNA replication, which is a necessary precursor to mitosis that occurs during the S part of interphase. Yeah, so interphase, the G1 phase. So immediately after cell division, the interphase begins with what is known as the G1 phase. Yeah, so the G1 phase is really the a large portion of the cell's life. Really, the important thing that's going on and what we're referring to here is the G1 phase is the cell's metabolism function in the body. It's doing a lot of things that are not really relevant to cell division here. One of the big things that is occurring right here is that the next phase, the S phase, which is over here, is a period of rapid and large-scale duplication of DNA. And so one of the big things that's going on in the G1 phase of interphase, or the G1 portion of interphase, is that the cell is gathering the raw materials and the energy needed to carry out that division. So that means a lot of gathering nucleic acids, a lot of gathering energy and storing energy, because without all of that preparation, that duplication would not be possible. Over into the S phase. So the S phase is the so-called synthesis phase, and during this, so this is the portion of the cell cycle when DNA is actually replicated for the production of two daughter cells from that one initial cell. So, of course, during the G1 portion of interphase, the cell has enough DNA for itself. There is basically one cell's worth of DNA within this cell. But of course, if it's going to duplicate at some point, it needs to have two cells worth of DNA within that one cell, which can then be partialed out to the two. This S portion of interphase is when that actually occurs. So basically, the cell gets all of its DNA, replicates everything. Basically, it takes the book of its genome and photocopies the entire thing, and then it does that in preparation for the creation of two daughter cells from that one initial cell. Um, yeah, and so I think this is a good point to talk about something that I personally have actually found confusing in biology courses in the past, and I don't think it was explained well, and presumably you'll do too if you've encountered this before. So yeah, prior to S phase, each chromosome has only one chromatid, and so these guys right here are chromatids. So here on the left, you can see that each of these two chromosomes has one chromatid. That's what it looks like basically before the S phase. And then in S phase, the chromatids are duplicated, and then each chromosome actually has two chromatids connected at this, what's known as a centromeric region, or a centromere. When you see photographs of, when students see photographs of chromosomes, generally speaking, what they see is actually this on the right. And that to my mind, is actually somewhat misleading, because that is a chromosome that has two copies of every gene, and it takes that arrangement very much in preparation for cell division. And so it's easy to get confused thinking like, are these this are these two um, arms, I guess, are they the same thing? Are they different things? That was actually never really explained to me. And but the reason why this is always shown, this here on the right, is always shown as an X rather than what it would look like before S phase, basically, little rods or hot dogs or something like that, is that DNA condensation occurs following S phase. So basically, before the cell divides, all of the genetic material is just kind of loose so that it can be used. And then in order for division to occur, it has to be condensed down into these chromosomes, which which are basically durable enough that they can be shuffled around during cell division. If there were DNA condensation to occur before S phase, what you'd see is basically 46 of these little rods or hot dogs, and I think that would actually, hopefully, having such a picture would belay some of the confusion about the structure of chromosomes. Unfortunately, that condensation doesn't occur, so we always see these X-shaped chromosomes in textbooks because that condensation occurs um, following division. Hopefully that makes sense. Let me know if it doesn't. Okay, so this figure depicts the cell during G2, immediately following the S phase. Note that before the S phase, there was a single centrosome, the centrosome, which are these guys, or these guys right up here. Um, so here there are two centrosomes, each consisting of two of these little rods or tubules. Um, those before S phase, there was only one of those. Um, Part of what occurred during S phase was actually the duplication of the centrosome itself, and these play an important role in cell division later. And then note also that during the G2 part of interphase that the chromosomes are still just dispersed genetic material. There aren't visible chromosomes that you could actually put under a microscope and see. And yeah, so another look at G2. Again, G2, the G1 and G2 phases of interphase are really the time when the cell is just carrying out its functions in the body. These are not immediately relevant to cell division in most cases. And so, yeah, the cell continues cell growth, but note that in the G2 phase, the cell now has 2 cells worth of genetic material in it. The mitotic phase consists of 2 subphases, mitosis itself and cytokinesis. Mitosis is the process of taking duplicated DNA and organelles and basically shuttling them to different sides of the cell. So you have, initially sort of a cell, a circular cell, with the DNA and the organelles just dispersed within that cell. During mitosis, the cell is preparing to divide, so all of the genetic material and all of the organelles get shuttled over to either one side or the other of that cell, so that then basically the cell can be cut in half and each one of the daughters can have everything that's necessary for life. Okay, moving into a description of the specifics of mitosis. So here we have the G2 of interphase. Again, that's not mitosis, that's the step which immediately precedes the beginning of mitosis. So what happened in, I guess, what has already occurred by the GAP2 portion of interphase is that the centrosomes have been duplicated, and then the chromosomes have been duplicated. So there is enough DNA for two cells within this one cell, and then the centrosomes can then be used for basically defining one side versus the other of the developing of the cell, of the daughter cells. Moving into prophase. So in prophase, the DNA all condenses down into actual chromosomes. So when you see a photograph like what I've been showing, you have all of those, of the 46 sort of X-shaped human chromosomes. That was probably taken during prophase, or at least during that period of DNA condensation. And you can see those condensed chromosomes here as these sort of bluish lines. And then what also is going on is that the centrosomes are beginning to migrate to opposite sides of the cell, and then are also starting to put out the tubules that are then used to very, literally physically drag the chromosomes apart, and then drag them to opposite parts of the cell. And then also during this phase, the nuclear envelope, that is the membrane surrounding the chromosomes, is breaking down because basically the chromosomes have to be taken elsewhere in the cell. The nuclear envelope, that membrane, is just simply no longer necessary. In prometaphase and metaphase, so the nuclear envelope is totally disintegrated. The chromosomes are just loose in the cell, and what happens is that the centrosomes have already lined up on either side of the cell. Basically, now there is one end or one pole, I think, as they're typically referred to. So there's one pole and the other pole of the cell. Those are basically going to be the centers of the two daughter cell once the cell is cut in half. And then in prometaphase and metaphase, these chromosomes line up in the middle of the cell. And so that basically allows them to be drug apart, very, literally physically drug apart by the tubules. In anaphase, this is basically that process of breaking the chromosomes into their constituent chromatids, which then become the chromosomes of the daughter cells, and physically dragging them to opposite parts of the cell. And then telophase and cytokinesis is basically finishing up the process. In this process, there is cytokinesis specifically refers to the process of cleaving the cell in two. And so you can very literally think of that as, as I've said, one soap bubble pulling off into two. And then each one of those soap bubbles is going to have a full complement of DNA, a full complement of organelles, and then will develop to a mature cell itself. I will not show you all this video right now because y'all can go watch it. This is a really, really cool microscopic video of a cell undergoing mitosis and cell division. Really quick, little short. It's a time lapse. I'm not sure exactly how much faster than real life it's going, but it would be hundreds of times, I would imagine. But definitely worth a watch. But I won't play it here. Or will I? No, I guess not. Meiosis. Okay, so we have been talking about mitosis, which is the division of cells to produce two genetically identical daughter cells. That, again, is the sort of standard or most common and abundant type of cell division. That's how we all grew from infants to adults. That's how our wounds heal. That's how our organs maintain themselves. That's really the bulk of cell division in the body. There is a different name for cell division resulting in what are known as gametes, basically sperm and eggs, and so meiosis produces instead of producing two genetically identical daughter cells, it produces four genetically distinct gametes, each with only half as much DNA as is present in a typical cell. And so each one of these gametes, or I guess, your cells, my cells, our main body cells, they have one chromosome from the mother and one chromosome from the father. These four gametes, they only have one chromosome. They have either the chromosome from the mother or the chromosome from the father. And of course, that's how, when, gametes, of course, undergo fertilization. When fertilization occurs, when the sperm and the egg meet, the offspring then, completes the cycle, and they have one chromosome from the mother and one chromosome from the father. And I'm not really going to get into the specifics of this because the actual mechanics of it are very similar to mitosis. I already kind of walked through what's actually going on at the cellular level, but I want y'all to understand that distinction, that meiosis produces four gametes, four sex cells, either sperm or eggs, and that each one of these cells has, has a chromosome only from the mother or from the father for each of the chromosomes present in the animal. So, we would have 23 for us, whereas our main body cells would have 46 chromosomes. And yeah, so I'm actually not going to go through all of the review questions myself. Unfortunately, as you all know, class was postponed today. But go over these. Make sure that when you study, use this as a study guide, because you may or may not see some of these verbatim or very close to verbatim on the test. And that's all I have to say about that. Please get in touch if you'll have any questions for me, or any concerns or anything like that. Have a fantastic day, and I will see y'all on Wednesday.